forward to the next hour. As I was saying, it's unusual for me, an unusual experience for me to give a talk where I'm not standing in front of a bunch of people and using my lecturer voice, but we'll see how this goes. I think that all of us will agree that object constancies are fundamental concepts in theories of perception. Constancies of color, constancies of shape, and of course constancies of size constancy, where we tend to see objects as being the same size independent of their viewing distance, at least within a certain range. And we can see this, I think, illustrated well in this photographic realism painting. At the end of the 19th century, photography was really capturing the attention of a lot of painters, and they attempted to paint as though it were a photograph. Here is a painting by Gustave Caillebotte in A Rainy Day in Paris. And I think you'll agree, the people here who clearly, in terms of the retinal projection, are much smaller than the people in the foreground. Nevertheless, you see them as being the same size. But if we were to take those individuals and clearly move them down here, now they look very tiny indeed. But of course, people are familiar, bananas are familiar, so it's possible that all of this depends on familiar size, but of course we know that's not the case. So even objects that are novel, we tend to see them as in their real world size, not as they are represented in terms of the retinal image size. So we see this object, which is a block of clearly on the screen of some indeterminate size, but it looks to be the same size when it's moved off into the distance. And indeed, powerful distance cues can even override familiar size. And of course, there are many examples of this that one can find. Here's a good one where these are clearly familiar objects, and yet because of the very strong perspective cues that are in the picture, we see the car in the more distant position as being bigger than the car in the forward position, even though clearly on the retina they have identical retinal image sizes. And we can see this is not a trick. We can move a bar from the apparently larger car to the apparently smaller car, and we can see indeed that they are the same size in terms of the retina, not in terms of how they are perceived in the real world. So back in the 19th century, Emile Emmert made it very clear that one can quickly calculate the real world size of an object on the basis of its retinal image size and distance. And Emmert's law, of course, has been applied not only to after images, which is what he was talking about initially, but also to real objects. And I think all of this can be summed up here by this character played by Hugh Laurie, who clearly has some problems with size constancy. So if you'll indulge me, I'll play this now, and I hope you can hear it.
Yes, I always remember this spot because it was right here. This was the very first time that I saw my wife. I was standing absolutely here, and I remember thinking, what an incredibly tiny woman she was. Absolutely tiny. She was about that size. Tiny little thing. It was only weeks later, as I got to know her, that I realized that was, of course, because she was a long way away.
Right. So I think that captures much of what I've been talking about, that clearly as images vary in size on a retina as a function of the viewing distance, it doesn't matter because we still see objects as being the size they should be in the real world. So, of course, naturally that raises a question: what are the neural substrates of perceptual size constancy? How is it that the brain computes the real world size of objects from distance cues and from retinal image size? And of course, I'm sure as many of you know, there's a history of at least 20 years in which people have been looking at the role of primary visual cortex in size constancy and its relationship with recurrent pathways in other higher order visual centers. Now, What's the evidence? Well, one of the first and most striking studies was carried out in Dan Kirsten's lab, led by Scott Murray, who was then a grad student in his lab. And what they did was to work with the Ponzo illusion, in which these two balls, which have the same retinal image size, in fact appear to differ in real world size, so that the one that's apparently further away appears to be somewhat larger than the one that's close. And so what you can do with this is that you can examine the activity along the calcarine, along the retinotopic distribution of V1, and one can actually map out the eccentricity map in this retinotopic area of V1, and then vary the size, the real size of the ball, and look at what how the variations in the size of the object are played out in the on the retinotopic eccentricity map. So having done that, then one can have people look at the apparently larger object, which in fact has the same retinal image size, or the apparently smaller object, which has the same retinal image size as this one. And as Scott and Kirsten showed, what you find is that the apparently larger one shows more activation in more eccentric positions than the apparently smaller one, even though the activation on the retina is identical in terms of its extent. Now, that depends, of course, the change in size, apparent size, depends on a pictorial illusion, the Ponzo illusion. And with pictorial illusions, there's a lot of cue conflict. I mean, you realize you're looking at a drawing, it's a flat screen, stereo, emergence, accommodation is telling you that that thing you're looking at is entirely flat, but the strong pictorial cues, nevertheless, will convince you that the man lying across the railway tracks in this position here appears to be larger than the individual here, when in fact they're the same size. But as I said, it's not as effective as real world stimuli, because what we have here is an illusion which is about 10 to 30%, depending upon the illusion and the setting in which the illusion is tested, 10 to 30% effectiveness in terms of indicating a change in the apparent size of an object, depending entirely on pictorial cues where there is cue conflict. So how do you get around that? Well, one way to do it is to use after images, in which you project those after images onto the surfaces of the world. And when you do this, of course, what you get is a perfect transformation of size, so that when you project the after image on the wall, it appears to be larger than when you project it onto a piece of paper held on your lap. And of course, we all know what an after image is, and I think we'll enjoy ourselves. It's early in the morning for me. If you stare at this little cross here for about I don't know, let's do it for about 20 seconds or so. Stare at that little red cross. I'm eating up all of my colloquium time here, but I think it's interesting nevertheless. Okay, so you should have an aesthetic experience now of some kind. It's a fun after image, and indeed, if you're a Canadian, I may be the only one on the Zoom meeting who is a Canadian. You never know. Oh no, I recognized Terry there earlier. If you stare at the dot here, then you can have a patriotic experience. So there clearly after images are effective, interesting, and the internet is full of them. What we did was to create after images, and we had people in the magnet, and we asked them to project those after images onto surfaces that were placed at different distances from them while they were lying in the scanner. And of course, according to Emmer's law, then, the further away the surface on which the after image is projected, the larger it would appear to be. So here's an example of the stimulus we used. You can stare at the little cross, and then I'll show you a blank screen, and you'll experience the after image. But if there's a light surface near you, like a wall or a ceiling, you might also try to project it there, and then you'll see that in fact it changes size, as it should, with 100% fidelity, because of course it appears to be actually stuck on the surface that you're looking at. So look at that cross. And there it is on the screen. And now if you look at a more distant surface, you'll see it appears to be larger. And the transformation is a very high fidelity transformation in terms of the apparent size. So this is work that was led by René Sparandillo and Philippe Chouinard. René is now at University of Trento, and Philippe is in La Trobe University in Australia. They were both postdocs in my lab. And so what we did, as I suggested, was we had people line the magnet. We had these little screens that would flip down. And initially what we did was to present people with a bunch of little LEDs in a circle and induced an after image, having them look at that ring of LEDs for about 4 seconds. And then we flipped down the inducing panel, and they were asked to project the after image that formed onto this surface here that could be placed at different distances from them. And so clearly when it was close to them, the after image would appear to be small, and when it was further away, the after image would appear to be large. And when you do this, what you can see is that the activation, as you move from a near distance to a far distance, the activation in V1 increases in size, and follows the size of the after image, the apparent size of the after image. And you can see this unfold in real time. And the great thing about after images is that they take about 2 or 3 seconds to form after you turn off the inducing stimulus. They last for about 8 seconds, and then they fade away. So they have the same time signature as a bold signal, which is rather nice. And in fact, when you correlate, we had people in the magnet press a button when they were experiencing the after image, and to release the button when the experience ended. After image went away, and we found a correlation with the bold signal that was essentially 1. It was 0.98. It's a nice relationship between a stimulus and the length of a bold signal in this event-related design. So here's a little movie of a trial in which an after image is induced. Oops, sorry, back up. So here we induce the stimulus, the after image rather, with the inducing stimulus, and then the after image begins to appear, that little purple spot, which indicates its size. And what you can see is that there's much more activation when the stimulus is further away and extends further more anteriorly, when the stimulus is further away, the after image is further away than when it's close. So it's clear then that activation in V1 reflects the perceived size of a visual stimulus, not its retinal image size. And so V1 plays some sort of role in size constancy, probably as part of a recurrent pathway that involves higher order visual areas. But of course, it's not clear when the integration of distance information and the retinal image size takes place. It's not clear whether or not this takes time to build up in V1, or whether it's there from the get-go. And of course, we don't know much from monkeys, because most single unit studies of V1 almost never manipulate the viewing distance and the retinal image size at the same time. And so there's been very, very few studies that have looked at the relationship between viewing distance and retinal image size in single unit work. Now, there are some opportunities for computation of the real world size in V1, because there's a number of papers that show the cells in V1, in the lateral geniculate nucleus, the pulvinar, that these thalamic nuclei and V1 can be modulated by eye position and other potential distance cues. This work goes back into the 1960s. Here's one example. This is from a study by Lau and Friedlander, and you can see modulation of neurons in the lateral geniculate of the cat by eye position signals. So you can demonstrate the presence of inhibitory gain fields and facilitatory gain fields, which theoretically then could be used to adjust the activation one sees in V1 on the basis of this distance information. So rather than using fMRI, which really has such a slow temporal resolution that you can't use it to measure the onset of any change that reflects a switch from retinal image size to a real-world size representation of V1. We used EEG, and this work was led by Zhuang Chen, who is my postdoc, who's now at South China Normal University in Guangzhou in China, Irene, and Molly Henry, who's at the Max Planck Institute in Germany right now, running her own lab. So we used an EEG system. We recorded from early visual cortex. And the setup was like this. We wanted to manipulate the real world position of a stimulus. And so we presented a stimulus on the surface of a monitor, which could be moved into two positions, either further away from the observer or close to the observer. And the stimuli we used on the screen were simply black discs, and they could have the same retinal image size by virtue of the fact that you could have a near small one and a far big one whose sizes had been adjusted so that they had exactly the same retinal image size, although they were clearly different in real world size. And you could also have two that have very different retinal image sizes, but have the same real world size, both the small pair and the large pair. When you look at the EEG signal, oh, hang on, I just want to, here's a sanity check. In fact, people did rate them as being large or small, independent of their viewing distance. So we were confident that they were doing the right computation. And when you look at the EEG signal of a mean signal from 16 people, what you can see is that early on, there is a separation between the smallest retinal image, which is the far small stimulus, and the largest retinal image, which is the near large stimulus. And the ones that share the same retinal image size, that is the far near and the near small, overlap. But as time marches on and the ERP passes 150 milliseconds after the onset of the stimulus, what you can see is that now the signals group by the real world size. So the smaller perceived size, whether it's near or whether it's far, are lumped together, and similarly the large stimuli are lumped together, whether they're near or far from the observer. So this evidence suggests that early on, V1 is reflecting, perhaps as it should, the retinal image size, but then as computations are performed on it, presumably again from recurrent pathways from V1, or perhaps it takes time for these early computations to be realized, you get real world size emerging in the representation of the retinal signal in V1. So it doesn't occur right away. It takes time to develop, about 150 milliseconds, but it does seem that size constancy plays out in these early visual areas such as V1. And as I suggested, this is probably dependent on a lot of recurrent feedback from higher order areas, particularly with respect to the pictorial cues, which clearly require some some computation.
Well, can I ask a question?
Of course you can.
So this should be reflected in the specific electrodes from which you're measuring. In other words, if you have an electrode overlying, let's say LO, you know, high order areas, presumably you should see these different, maybe more stronger or more, or only they're selective for that. Have you compared different electrodes?
No, we haven't. First of all, we did do some probes, but we couldn't, with the setup we had, we couldn't see anything really interesting emerging. It was pretty noisy, but it did play out very, as you saw very clearly in the integrated early visual signals, but we couldn't see much further on, perhaps because it was harder for us. We didn't have any way of doing any source coding in terms of LO and so on in these individuals. But it's a great question, and I think MEG would be a wonderful way to go, where you could compare the fMRI activation with MEG signals playing out. And had we an MEG, that would be something that we might want to explore. And it may be something that Zhuan might do in Guangzhou, because she has some access to an MEG machine. But good question. Okay, so I'm going to change gears a little bit, because size constancy has been studied to death in perception. We know a lot about size constancy and how it operates, although we don't know so much about its neural substrates. But it's seldom appreciated that we also show size constancy when we grab things. So that when we are dealing with our coffee cup, whether the cup is further away from us or close to us within prehension space, we open our hand the same amount, and as we adjust our hand in flight to the size of the object we want to pick up, even though its retinal image can vary as it's nearer or further away from us. So how does grip constancy work? How does it depend on the same computations that we see for perceptual constancy, or is something else happening? So that was a question we began to address. And I have to take a little detour to actually flesh out the story. And of course, as some of you know, some years ago in the last century, we put forward the idea, we being David Miller and myself, the idea that two kinds of visual systems emerged in the visual pathways in the cerebral cortex. One that allows you out there in Zoom world to recognize the objects in that picture, the cup and the pencil and the wristwatch and the hand and so on. And that visual system is functionally and neurally independent to some degree, we argued, from the system that allows the actor in the picture to reach out and pick up the cup, for example. That fundamentally different transformations are involved in fundamentally different brain structures are engaged. And indeed, in a long series of studies in our lab and others, it seems that the pathway that runs from early visual cortex up into the posterior parietal cortex is important for transforming visual information into the required coordinates for action. And similarly, the projections that go ventrally down into the temporal cortex are important for transforming visual information for all of the cognitive operations that we might wish to perform on them. And of course, the two streams interact enormously in the control of actions directed at objects where hand postures could vary importantly, such as using tools and cups and so on. I think it's interesting the posterior parietal cortex is well connected with premotor cortex. It sends projections down to premotor neurons in the brainstem, and the infratemporal cortex projections are well connected with medial temporal areas and inferior prefrontal areas that are important for cognitive computations. So how did we come to say that? How did we come to have that idea? Well, the Rosetta Stone for us was a patient who's probably the best studied visual form agnostic patient in the world, patient DF. She's now in her 60s. We began to test her in her early 30s. And She was a woman who developed hypoxia from carbon monoxide poisoning. And in these much more recent structural MRs, you can see that she has bilateral damage to area LO, quite complete damage to area LO in both hemispheres. And of course, her most salient symptom is visual form agnosia. She can't recognize objects on the basis of their form, but her clinical and psychophysical testing in terms of acuity and so on is essentially in the normal range, some deficits at low spatial frequencies. So what happens when you ask her to reach out and pick up an object or to give a perceptual report to manually estimate its size by opening her finger and thumb a matching amount? It's an old story. We track these movements, and what we find is that when she reaches out to grasp objects of different sizes or different widths, she opens her hand wider in flight for the wider one as compared to the more narrow one. But when she tries to indicate the size of the objects, she's basically a chance with enormous trial-to-trial variability. And of course, she can't verbally describe what size it is either. She really doesn't see the width of the object, despite the fact that her hand scales in flight to the real size of the object. And of course, you can contrast that with patients who have damage to the dorsal stream. As a patient RV, a woman who had bilateral strokes separated by a week or so, with damage in the right hemisphere, in the posterior parietal cortex, and damage in the left hemisphere. And when you test her on the same kind of tasks, what you see is that she knows how big the objects are. She can verbally describe that. She can say, oh, that's the square one, that's the narrow rectangular one. And of course, she opens her hand. She's not quite normal, but she opens her hand to match the size of the object. But when she reaches out to pick it up, she just uses this default posture where she just opens her hand very wide on every occasion until she touches it. And then she closes her hand around the object. So it's a nice dissociation. It's the kind of dissociation that neuropsychologists love, a double dissociation, where damage in one place, the ventral stream, produces a deficit in perception, but leaves her grasping intact, and vice versa for damage in the dorsal stream, which leaves perception essentially intact, but interferes enormously with grasping. Okay, so I'm not going to belabor this anymore, but there's a lot of evidence from neurological patients that this division of labor in the cerebral visual pathways is a viable way of characterizing the transformations that are performed in these different visual pathways. There's lots of evidence from single unit recording in monkeys. There's more controversial evidence from visual motor psychophysics about whether or not grasping is less sensitive to illusions of size than our perceptual estimates. And of course, there's fMRI. Many papers now in both patients and in normal observers show that you can get activation related to grasping in the posterior parietal cortex and activation related to object recognition in the ventral pathways. Okay, so two visual systems. Now, the emphasis for many years has been on the fact that you get these projections emerging from early visual cortex into the dorsal stream or into the ventral stream. But of course, projections from V1 are not the only visual inputs into the two streams, and we have to remember that. This is a kind of model that almost all electrophysiologists and people doing fMRI work with, which is projections from the geniculate to the primary visual cortex, and then from the dorsal, from there to the dorsal stream or the ventral stream. But of course, there's projections from the interlaminar regions of LGN that project. There's projections via the colliculus and the pulvinar, and of course, there's all these other projections that have only been studied a little bit in primates. Most of the work, for example, in accessory optic tract has been done in birds and in rabbits, not in monkeys. And I know of no human work. And if you take away V1, there's clearly lots of opportunities for input via the middle temporal complex into the dorsal stream or to the ventral stream. So there's a lot of, there's a spate of recent papers that looked at these pathways. We know that there's projections that go from the interlaminar regions, the corneocellular pathways that project to MT as well as other visual areas, and from there project to the dorsal stream, a little bit to the ventral stream. There are projections that go to the sphere colliculus to the interlaminar region and then to MT, projections that go from the colliculus to the pulvinar. And there's also projections, they're much bigger in the infant, it's been studied most in the marmoset, that go to the pulvinar and from there to cells in MT, and from there to the dorsal stream. This pathway is less strong in adults, has not been studied in humans, and very little work in macaques. But marmosets, there's quite a bit of evidence for this pathway. So one can ask, what happens to perceptual constancy and grip constancy in people who have V1 lesions? And we had an opportunity to look at such a patient. This was a patient who, well, she's now, this is a bit out of date, this slide, but she's now 45. But the time we first began to test her, she was 40. And she basically had an iatrogenic problem. Her doctor, she went to the doctor with an aching shoulder and complained of this. And he sent her home with some Tylenol, but it turns out she had a lung infection and she developed severe hypotension, and she went into a coma for 2 months. And sometime during that period, she had a large bilateral occipital infarction. She's a wonderful person, has a great sense of humor. I've got to know her very well. But she's completely blind in static clinical perimetry. She can perceive moving objects. She has retics, so she can still see moving objects, and she can even report the presence of static high contrast stimuli. So, you know, a big stimulus that you might want to grab, for example, she can see that, and it's a conscious experience for her. So I'm going to show you her lesions, and if you've ever looked at these kinds of slides, I think you'll be rather shocked at how large the lesions are. So this is the lesions in her visual cortex, and they extend well into the ventral stream. This is a slice going through the temporal lobe, and you can see that there's really no tissue here. There's a bit of spared perihippocampal in the right hemisphere. There's a tag of very anterior calcarine subserving the very far peripheral visual field. And the lesion even extends into the occipital parietal area in the right hemisphere. So these are very big lesions.
Mel, may I ask a question?
Yes.
You said that she recognizes moving objects, and I want to ask, I mean, I understand.
She doesn't recognize moving objects. She sees movement.
Oh, she just sees them moving, but she doesn't understand what is moving.
No.
Okay, thanks.
There's a way, her daughter, who's now in her late teens, she was 2 when she had the stroke. She has worked very hard along with her husband to train her to read very large capital letters of the alphabet. But I don't believe that she can do this. So she will, if you put a big letter A on a piece of paper, she will look at it and she'll move her head back and forth in her eyes like this. And after several seconds, she'll guess correctly. But I think what she's doing is using some difference in the motion signal to recognize the letter. It's not a kind of percept of the letter A or a percept of the letter B. She believes that she can see colors, but she can't. We've tested her rigorously psychophysically on colors, and she cannot tell colors apart. But what she can do is this, and I'll show you.
Awesome.
This is not an experiment, this is a demo.
that's the weird one, but see, I never realised that until I caught it.
It's got it. Okay, so she can catch balls very well. Whoops. We tested her with static stimuli as well, and these are the so-called Ephron blocks developed by Ephron for actually testing patients with apperceptive agnosia, visual form agnosia, where they, what you have are objects that have exactly the same surface area, but vary in their dimensions. Can you tell them apart is really the question. And this is work that was carried out by my then graduate student, now a postdoc with GenEnds, although he's still working in my lab and working long distance at UBC. And he presented these stimuli as pairs and asked her whether they were the same or different. I mean, it was the world's simplest discrimination. Are these two objects the same or are they different? And basically, she's a chance. She can't tell them apart. So we thought, well, what about estimating the size? Could she estimate the size with her hand? And of course, then we also tested her on grasping. So these are her manual estimates. And basically, she's flatline. She can't tell you the size of these objects. But now when you ask her to reach out and pick them up, things tighten up. And she shows a slope. That is, she opens her hand wider for big objects as compared to small objects. It's not a normal slope. It's not as steep, but it's very reliable. That is, we've tested her in other settings as well. She opens her hand wider for larger objects than she does, for fatter objects than she does for narrow objects. So that suggests that despite the damage in V1, her visual motor system is somehow supporting grasping that respects the dimensions of objects, even though she can't report their width. So what's going on here? One way to find out whether she has any kind of normal size constancy, grip constancy, is to vary the distance. And in those studies I've just shown you, we didn't do that. They were always presented at the same distance. So there's just a possibility that she was using some cue from the size of the retinal image, and she could remember where the distance was, and somehow that's how she could scale her hand to the object. So we really asked the question, does she show grip constancy when grasping objects placed at different distances? And again, this is work carried out by Rob, along with help from Irene, Gavin Buckingham at Exeter, who was then in Scotland, and Philippe Chouinard. The reason so many people are involved is that we use different labs, both here and also in the UK, to test her. She lives in Glasgow. So we did a perceptual matching task in which we varied the distance of the object that she was to estimate with her, by the opening of her finger and thumb, a kind of magnitude estimation task, if you like, but using the same effector that she was going to use to grasp the object. And as I said, we know she scales for object size, but what about object distance? Does she open her hand the same amount independent of the distance the object is away from her? And the answer is no. So although the controls show great grip constancy, opening their hand the same amount over distances from near to far, she opens her hand wider for closer objects, which of course have a larger retinal image size than objects that are further away. Did I say, yeah, so this is for matching. So she matches incorrectly. She matches, and actually quite reliably, to the image size, not the real world size of the object. So what happens when you test her now on grasping? Well, what happens is very striking. She's normal. That is, in flight, she opens her hand to the real size of the object, independent of the viewing distance of the object is. And she also shows all of the normal changes in velocity that one would expect to see for objects that are closer or further away, which I haven't got time to talk about right now. So basically, in this sort of summary abstract here, this is a person who has bilateral lesions of primary visual cortex extending into and including areas in the ventral stream, such as LO, who shows estimates that are based on retinal image size, but her grip apertures reflect the real world size. So an interesting dissociation. So it indicates that perceptual size constancy and grip constancy depend on different visual pathways, with only the former involving V1. So there's someone talking, so I thought maybe they're asking a question, but that's okay.
Yeah, I muted that.
Yeah. So these extra geniculostriate circuits involve projections from the pulvinar probably, or perhaps the LGNd. We tend to favor the collicular pulvinar route, or even the route from the eye to the pulvinar, and they theoretically could compute the size of the goal object and/or, this is important, the spatial locations of these stable grasp points on the surface of the object. That is, it may be that the computation involves putting the index finger, in normal people as well as her, in one location and the finger in another, which would also show grip constancy. So it's, in any case, it requires that one map out where to put one's fingers according to an adjusted computation based on retinal image and the distance of the object.
Mel, sorry, may I ask another question?
Yes.
It's actually not exactly my question. Avi Portnoy is asking whether it may have to do with convergences, and let me read precisely.
Sorry, I just want,
Avi?
Sure, go ahead.
Back a bit. The z-axis, would that trigger an accommodative or vergence response?
Yes, we didn't measure eye movements. She's got poor eye movements, so it could be that vergence isn't an important cue. It could be accommodation.
No, okay. I didn't mean specifically to her, but I asked it at the beginning of the lecture when you were showing some of the participants in the magnet, when you were able to. They were.
Oh, I see. Yes, yeah. When you look at the, you're absolutely right. That's a worry. So is it simply that you're verging eyes in a different position, and that's to do with the signal? What we did was we moved fixation points in the supplementary material in the article. You can read about it. We also used real objects, real 2D objects that we placed at different distances. It's to do with the size of the object, not the vergence signal.
Okay,
good point.
Thanks.
Yeah, but I just want to ask whether maybe these aspects, or her dominant eye, or the fact that maybe these also can serve as different cues. And when she's actually using or preparing for a grip, she adjusts some of that, but it's not happening when just maybe
Sure.
the perception, just the dominant eye is.
Yeah, whatever it is, it's a signal that doesn't involve V1 because she doesn't have V1.
Mm-hmm.
So yes, and it looks as though, and you may be right, that information can be used by the visual motor systems that are controlling grasping, but clearly can't be used for perceptual report. Absolutely.
Thank you.
So most recently, I've been working with a group in Australia trying to develop a potential animal model for studying the role of, in this case, pulvinar circuits in size constancy. And we've been looking at grasping in the common marmoset, which does grasp objects, particularly in a feeding situation. It'll pick up pieces of papaya or banana or other things very adeptly, and then obviously convey that object to the mouse. And here we see, this is work that was initially carried out by, after a lot of discussion on Skype between myself and James and his graduate student Dylan Fox, developed a system for looking at grasping in the marmoset. And here we can see the marmoset sticking its hand up. We have pieces of food here, papaya, that are different sizes. And we have a proxy for this whole hand grasp, which is the finger and thumb. And we can trace out the trajectory here. And what you can see is that There's this normal overextension of the fingers and then the closing down on the object, which one sees in humans, either in a precision grip or in a whole hand grip. And we can see this obviously only in a whole hand grip in a marmoset. It doesn't show precision grasping of these objects. And when you compare, you can force the animal to use its right hand or its left hand by having a hole in the, which it reaches through, which is hard up against a wall either on the right or the left, and so it's forced to use either one hand or the other. Dylan, I have to say, is a marmoset whisperer. He can really work with these animals and get them to do some amazing things. So when you do this, what you see is this is the left and the right hand. There is scaling for object size, so that they open their hand wider for big pieces of food as compared to small pieces of food. So it's a nice model. There are differences between the left and the right hand. James is convinced they're reliable. I'm not so sure. Partly there's a problem that there's slightly different camera angles for the left and the right hand, and you're never sure exactly what the whether these distances between the finger and thumb are real or not. They certainly are real in terms of the object size, but whether the comparison between the left and the left hand and the right hand is real is not clear. So in our initial attempt, this is work led by a postdoc in the lab in Monash, an MDA lesion of an infant monkey in the inferior part of the medial part of the inferior nucleus of the pulvinar, where we know it receives input both from the eye and from the colliculus, and then projects up to MT. That pathway has been well studied by James. And when you do this, then what you can see is the pathway that then goes from MT up into the posterior parietal cortex on the lesion side is entirely, just doesn't appear. It's well formed on the control side, but absent on the lesion side. And then when you look at the actual size of the grasp, what you can see is the grasp has many changes in its velocity and acceleration profile. And also, basically, the animal reverts to using a very large grasp when it reaches out to pick up the object, as opposed to a grasp that's well tuned to the size of the object. And we can see this in the movie here. Control hand and the lesion hand, different marmosets. But you can see this very wide grasp on the lesion side. The fingers are splayed compared to the control animal. So these are infant monkeys, and so clearly things the lesions were done when they were infants. They're adults here, and clearly this is a developmental story. Future work then, we want to examine the effects of the inactivation of the pulvinar and the LGN separately on size constancy perception and grip constancy in adult marmosets, and that's work that's ongoing now. So where am I? I have a tiny bit left. Is it okay to carry on? I can't hear you, Sharon. You're muted. It's okay?
Sorry, yes, I said we've got 10 more minutes.
Okay, that's fine. I'll be there. I'll be fine.
And we're not in the middle of semester, so okay. It's not a problem to go further. Yeah.
All right, so the summary so far: perceptual size constancies depend on re-entrant processing involving V1. At least that's what we believe, and certainly the evidence points to that. Grip constancy, however, seems to be mediated not by these projections, but by other ones that to the dorsal stream that are independent of V1. So I want to raise another point, however, and this is that there's a big difference between size constancy for perception and size constancy for grasping, in that grip constancy normally operates in peripersonal space, where you can reach. Whereas in contrast, perceptual size constancy, it extends many, many meters out into the world. And so you could expect, therefore, that the computations underlying grip constancy, they could incorporate distance information from proprioception, whereas that wouldn't be the case for perception necessarily. So that when you lean forward, you get proprioceptive information about where you are with respect to the object. When you're holding something in your left hand and you grab it with your right hand, you get information from proprioception. And so in a recent study that was led by Joan and Reni, we used some glow-in-the-dark spheres to look at this. And so we put these spheres separately on different trials on 3 different locations. And people either held onto or didn't hold the pedestal. So the pedestal was always the same size. But of course, when they were holding onto it, they had proprioceptive information about how far away the object was, glowing in the dark. They couldn't see their hand, they couldn't see the pedestal, but they could see the glowing ball, and then they could grab it. And so I'm going to be comparing the 25 centimeter wide and the 50 centimeter or millimeter wide objects. So when they're either holding the pedestal or not holding the pedestal, and they made a grasp beginning here and then moving over, or they did an estimate of the size of the object. And the viewing conditions were such that they did it with full binocular vision in the light, or pinhole viewing, where they looked at the glowing ball through a pinhole. And of course, that eliminates vergence, accommodation, pictorial cues, stereopsis. And all you see is a glowing ball. So most of your distance information, if you're holding the pedestal, will come from proprioception. Okay. And so it looks something like this to set up. Over top of the pinhole glasses, they wore translucent LCD glasses that we could then make transparent electronically. And it went something like this. So there's a grasp. Or a manual estimate. I think it's this big. Then we gave them feedback. So they had feedback on both manual estimates and on grasping about the real size. So if they were off, they could correct theoretically on the next trial in the manual estimate. Okay, so here's their manual estimates, both near and far. And what you can see is they're overlapping. They open their hand wider for the big object compared to the small sphere, and there was no effective distance. They were showing perfect size constancy, whether they were holding the pedestal or not, as one might expect in their manual estimates. Now, when you gave them restricted viewing, where they were looking through a pinhole, they saw a glowing ball, and when they were holding it, they had information about how far away it was. They didn't seem to use it very well. They still saw the ball as being larger when it was closer than when it was further away. So let's look at grasping. Well, under full viewing, not surprisingly, they scale their hand in flight to the real size of the object independent of viewing distance. And when they grasp it in restricted viewing, where they have virtually no cues to distance except proprioception when they're holding it, now you see a correction. That is, they get a return to size constancy, or sorry, to grip constancy. But when they're holding it, of course, they don't show grip constancy. They open their hand wider for closer objects than closer spheres than far away spheres in this glow-in-the-dark situation when they have no distance cues, and no proprioception. So proprioception then will correct for these deficits in visual information, distance cues in this situation, but not for manual estimates, not for perception. So the bottom line then, holding the pedestal on which the sphere sits restores perfect grip constancy, but not perceptual size constancy. And it suggests that proprioceptive cues to distance can compensate for these unreliable visual cues when people grasp objects, but not when they estimate the size of those objects. Now you might ask, well, why on earth wouldn't you use it in perception when it's there? And I think it's because the system normally has vision throughout, including peripersonal space. All the computations that are done, whether they're further away or whether they're close, can use these very reliable visual cues. So the bottom line of the whole talk then is that perceptual size constancy and grip constancy appear to be mediated, I hope I've begun to convince you, by quite separate neural networks, with the former but not the latter, depending upon these geniculostriate projections. Thanks very much. That's it. I'm done.
Thank you very much, Mel. And I'd like everybody, well, I'm inviting everybody to unmute yourself, and let's give a big applause to Mel. I wonder if I can unmute everybody. Thank you very much. I hope you have time to answer questions that I expect.
No, I gotta have breakfast now. No, I'm fine.
Okay, so everybody feel free to unmute yourself. And I just want to say before we start the questions that next week we have Daphne Moore, who will speak about how baby sees. And I wrote in the chat our website. I put down our website so you could go in there and have a look at our next Tuesday talks throughout the year. And yes, everybody's welcome to ask questions. Thank you very much, Mel.
My pleasure.
May I ask, Mel?
Yeah. Oh, hi, Lauren.
Hi there. It's a very nice story and great data, but I'm a bit skeptical about your arguments about V1. It seemed to me that the,
what particular part of V1?
Experiment seemed to give you some hold on V1, but I was not convinced that the EEG experiment does. You don't really know where those waves are coming from, particularly the later ones, and the idea they're coming from V1 rather than some extra striate area seems to be rather thinly based.
Sure. Well, they have to, early on, I mean, they have to come from V1 in the sense that in order to get to the higher order visual areas, they have to come from V1. But given the position of the electrodes and also the C1 wave, which, by the way, I didn't discuss at the very, very beginning, which is most people agree is very thoroughly a V1 signal. It shows retinal image size computations.
Oh.
And those early visual signals, yes, we can't pin them down to V1, but those early visual signals would appear to reflect the retinal image size, whereas later they are converted into the real world size. Exactly.
Yeah, that makes sense. But I thought you were trying to argue that V1 itself was modulated by in a size constancy kind of fashion.
Well, it is from the fMRI data, and so I'm assuming that what we see in the EEG would map onto that fMRI data. In fMRI, V1 along the calcarine shows real-world activation. That is, it gets more eccentric the bigger you think the stimulus is, whether it be in a Ponzo illusion or whether it be an after image projected on different surfaces. And the EEG signal, admittedly, it can't be pinned down to V1, but it does show that that doesn't emerge until much later.
Can I follow up?
Yeah.
It's a follow-up on this question and the fMRI. It will be useful if you could actually do a physical or optical simulation of the size constantly change. In other words, I would like to see the fMRI activation in the same subject, for an actually enlarged ring or whatever, and to compare.
Yeah,
and
it's in the supplementary material. In the paper, we did it.
And it compares precisely
It compares precisely with what we see with the after image. We began with the after image because we thought it was sexy, in the sense that it is something that is stuck on the retina and then can be projected onto surfaces. But then one of the reviewers asked us to do real objects, and we did.
Okay.
Maybe that was you, Rafi.
No.
I see that there is a question in the chat, so I will read it to you. It says, following up on the pinhole experiments, can I assume perceptual size constancy is worse in monocular peripheral visual field, but perhaps grip constancy would not be impacted as much?
Yeah, that's an interesting question. Because of course, grip constancy, the cues that are used in grip constancy in the real world probably depend a lot on things like vergence and accommodation, which of course we eliminated with the pinhole viewing. And it was, fortunately, what happened was that the proprioception information actually substituted for the removal of accommodation and vergence. In the case of the case of perceptual size constancy, I think that pictorial cues are incredibly powerful. I mean, if you cover one eye, the world doesn't change. I mean, it looks remarkably the same, and size constancy continues to operate. So I think that certainly at distances beyond a few meters, vergence and accommodation and stereo are probably not as important as pictorial cues for perceptual size constancy.
Okay, thank you. That was from the chat.
Can I just ask a silly question?
Sure. I can give you a silly answer.
I've always been puzzled as to why, looking out of a plane window, you lose everything about size constancy. It looks like tiny Toy Town if you're
very
relatively low. And I would have
I
thought you have plenty of information to know what size things are, except if you're in something like the painted desert, which is very confusing in terms of size constancy because you don't know how big the mountains or hills are.
Yes, I think size constancy beyond several, let's say beyond 60 to 70 meters, probably depends more on things like familiar size. I think we really have very little idea of the size of objects I mean, we have a good guesstimate that that's a bloody big thing, a mountain, but I don't think we, big changes of hundreds of feet in the size of the object would not register, I don't think.
Yes, but we know how big houses are.
Yeah, exactly.
As tiny houses. So some part of the neural system must absolutely pack up on being able to use anything but the retinal image.
Yeah, so I guess size constancy in vistas, which would be the only kind of comparable thing that would be around when we were evolving our visual system. We weren't in airplanes. I think maybe the need for a good deal of sharp computation for size constancy was probably not that great, and we would depend instead on familiar size of antelopes and other things that were bounding across the veldt. I mean, that's just a just so story. I have no idea why things fail at very far distances.
Mel, I'd also like to ask about V1. I mean, assuming that V1 is mainly retinotopic, and so when it is influenced, even at the second or, after 100 and 200 milliseconds, if it has some feedback or is incorporating more information than the ones that are coming from the retina, and is actually changing its activation to reflect size constancy or world size rather than retinal size. So I'm wondering if you think that maybe what is being measured may have to do with the error or prediction error, or maybe it's not as retinotopic as we've anticipated or as we assumed. I mean,
yeah, it's a wonderful question as to why it plays out that way in V1, both in the fMRI study and the EEG, and not just my lab. There are many labs now that have shown that V1 seems to change either over time or in fMRI study you can see it with changes in the real world size of objects where the retinal image stays the same. Why that happens is a wonderful question, and whether it has to do with prediction error and things of that kind is a possibility. I don't know, and it's a question that's emerged late in the game, I think, because although people were studying retinotopy for years in single cell studies and also laterally in the magnet, there's never been much manipulation routinely of distance and retinal image size in a way that you can unpack that story. So I think it's ripe for investigation and understanding theoretically what the hell what's going on there in terms of the real world computations that are apparent in V1.
If I can make a comment.
Go ahead, Rafi.
You know, fMRI is very sensitive to top-down attention effects, spotlight of attention, much more than neurophysiology. It might be related to attentional modulation, this after image effect in V1.
Sure, but it's related to the real world size.
Yeah,
so it's not as though you're attending to some other aspect of the scene.
No.
You're attending to the edges or size of the object, right?
It nicely shows that attention is actually real world rather than retinal image.
But if you manipulated the salience of the target in some way that didn't involve changing its size, you think that might produce equivalent changes in the bold signal?
I see. So you think that if the target remained the same real world size but became much more interesting and you wanted to attend to it better, you might show some increase in the eccentricity of activation in V1. Is that what you're arguing?
I'm suggesting that's some kind of control, yeah.
Yeah, possibly. I don't know. It seems unlikely to me, but possible.
Hi, Mel.
Oh, hi, Paul.
Hey, how's it going? I just wanted to ask about MC's grip constancy.
Yes.
So she can also, I don't want to use the word see, but she can also see her hand grasping for the object. So would you expect her grip constancy to be the same if it was blind grasping?
So what we've done is, in a separate experiment I didn't discuss, in which she does show grip constancy, continues to show grip constancy, we used phosphorescent balls, and but what we did do is to provide her with an image of her hand by making the glove phosphorescent. So you're right, she saw the hand as she was reaching out to grasp the object. But I think it would be a stretch to say that somehow she was using some kind of a primitive perceptual system, not involving V1, to somehow look at the grip size as she was moving towards it, largely because we know from work in normal neurotypical individuals that the maximum grip aperture is achieved very early on. Like, it's programming that occurs. It's not as though you're adjusting your hand in flight, matching your grip aperture to the size of the object. You only do that in a normal individual in the very closing phase. You don't do it when you're reaching towards the object. And in fact, you build in an error factor when you're doing an open loop grasp where you can't see your hand or the object. You open your hand wider to build in the air. So I don't, for many reasons, I don't think she's, if that's your question, maybe I didn't let you get to the end of it, that she's comparing her hand to the object.
Sure, thanks.
Another question, I don't know if it's going to be the last one, but first of all, there's one I see now in the, from Lauren Murray. He asked, or she asks, could perhaps the size constancy issue when viewing objects from high up, an airplane, for example, be affected partially by height and field cues? Objects lower in the visual field tend to be closer, not further away.
That's right. That's right. That is a distance cue. It's not totally reliable, but absolutely correct. That is, things in the lower visual field tend to be closer to you than things in the upper visual field. And of course, people have talked about that in the context of visual search and so on, that you are, you search for objects more in the upper visual field than you do in the lower, perhaps because things are much more likely to change out there.
Yeah, in the upper visual field.
Yeah.
And I've
got yes, so I could, because when you're looking out of the airplane window over the cliff, then clearly the lower visual field is now very far away, which is unusual. Yeah, that's true.
And the clouds tend to look bigger.
I don't know, I've never measured them, but they certainly look big.
Yeah. I have another question. I mean, I've not done electrophysiology, but my assumption is that most of the electrophysiological studies, let's put aside Rizzolatti with the grasping, but many of what we know about the visual system is actually from short viewing distances, similar to human studies. I don't know if that's correct. You can
yeah, that's true. I mean, I guess typically because of the Octan law, monkey physiologists have placed their stimuli about 57 centimeters away from the eyeball, which means that 1 centimeter is 1 degree. And so that's kind of convenient for doing your computations. And that's how most monkey electrophysiology has been done, and cat electrophysiology for that matter. There are people like John Ullman and others have very distant systematically and have discovered nice modulation of cells in V1 and other areas of the visual cerebral cortex that are modulated by distance.
So this actually, your findings in a way challenge a lot of what we know about visual system function or organization, taking into account that, a human lab psychophysics and MRI is also from quite usually from a rather fixed distance of not more than a meter away.
Yeah, exactly. So it could be that let's talk about monkey electrophysiology for a moment. It could be that people look at the early and late spiking in a signal from a neuron, say in V1. And they characterize it, but you don't know that if you were to move that stimulus closer or further away, the later components would look very different. You just don't know. I mean, because it hasn't been done, except by people, as I said, and not very little work by people like John Ullman. It's not a routine thing that's been done throughout the visual system. And the same is true of fMRI.
Yes.
Now, there are people who have looked at size constancy, obviously, but they, I think, strangely have been using images of objects that of familiar size that could be bigger or smaller. It's unusual to move things actually further or closer to people.
Thank you very much. Yes.
Is there time, Sharon, for another question, or is it too late?
From my point of view, everything's okay, but Mel has to decide.
Oh, no, I'm fine. Yeah, go ahead.
Hi, Mel. First,
hi, how are you? Is that Patricia? Oh, Patricia. Is that you? Yeah,
it's me. Thank you for the great talk. I have a comment on the grip constancy that I agree with you. It depends on pathways that pass through subcortical structures to the posterior parietal cortex. And I would like to remind you that also other routes, in addition to MT, are reached directly from the medial pulvinar nucleus. For example, V6A, we showed in the macaque, of course.
V6A, right.
It has a direct input from this nucleus that you injected with James Bourne. And so not only MT is a candidate through which the information can reach the posterior parietal cortex for allowing grip constancy.
Right. So there are direct projections from medial pulvinar to V6A?
Yes. Yeah. Yes, we found them directly.
Yeah, the pulvinar in the macaque, as you know, and in the human is the largest thalamic nucleus, and there's so many different subregions, and we understand so little about what it's doing. There are experiments, neuropsychological experiments in humans with pulvinar damage that show that they have deficits in reaching, which is interesting.
Also in the monkeys, this has been shown by Melanie Wilkie.
Yes, Melanie Wilkie, yeah, exactly. She's also done it in humans, exactly.
Yeah. Thank you very much.
Well, thank you very much, folks. That was great.
So
Really enjoyed it.
Yeah, let's again give a big applause for a wonderful talk.
And Rafi, I guess I'll see you next week.
Yep.
All right. Okay. You take care.
Shana tova.
Thank you very much.
Thank you.
Bye-bye.
Bye.
Bye-bye. Bye, Mel.
Thanks, Mel.
Bye-bye.